picture two people, right? One eats burgers and fries like every single day, never steps foot in a gym, and yet has perfectly textbook cholesterol.
Yeah, we all know someone like that.
That was so frustrating. And then the other person eats steel-cut oats, runs marathons, actively tracks every macro, and is still staring down the barrel of an early heart attack.
Right, which feels totally unfair.
Exactly. But today we are looking at a massive body of research that proves it is not just, cosmic bad luck. The answer is actually hidden in the absolute rarest typos of our DNA.
It is, it's an incredibly precise science. We are talking about minute single-letter changes in the genetic code.
Like almost invisible changes.
Completely. Changes so microscopic they're barely there,
Yeah.
but they trigger these massive cascading effects throughout the entire human body.
And to figure out exactly how these typos work, we are jumping into a really monumental source for today's deep dive. It is a newly published Nature Genetics paper from May 2026.
Yeah, a truly landmark paper.
So it's a comprehensive exome-wide association study, and it is focused entirely on blood lipids. And our mission today is to explore how scientists managed to analyze the genetic data of over 1.15 million diverse individuals.
Which is just a staggering number of people.
Right. And they're hunting for incredibly rare genetic mutations that act as, master switches for our cholesterol and triglyceride levels. So, okay, let's unpack this. How do you even begin to look at the genetics of over 1 million people?
Well, the sheer volume of data here is what makes it unprecedented. We are looking at a level of genetic resolution that simply did not exist even 5 years ago.
Because to find something incredibly rare, you need an unfathomably large haystack.
Exactly. So you have to combine forces. The researchers aggregated data from 3 massive ongoing genomic initiatives.
Right. They pooled the VA Million Veteran Program, the UK Biobank, and the All of Us Research Program, right?
Yep, those 3. And when you put all of that together, you get 1,158,017 individuals.
But looking at the source, what really jumped out at me wasn't just that final massive number, it was who actually makes up that number.
Oh, absolutely. Historically, genetic studies have relied heavily, almost exclusively, on populations of European descent.
Right, which leaves huge gaps.
Huge gaps. But this study completely breaks that mold. A crucial 20.57% of the participants, so that is over 238,000 people, come from non-European populations.
Wow.
Yeah, we are talking African, admixed American, Hispanic, South Asian, and East Asian ancestries. And we will get into the mechanism of why that completely rewrites the rule book later, but, it is the foundational strength of this data.
Okay, so the specific targets they were tracking across all 1.15 million people were their blood lipids, right? So total cholesterol, low-density lipoprotein, high-density lipoprotein, and triglycerides.
Standard lipid panel.
Right. Now, they specifically conducted an exome-wide association study, so let's make sure we are completely clear on what that means for you listening before we go hunting for typos.
So if we think of your entire genome as a massive, towering library of recipe books for building a human being.
Okay, I like that visual.
Yeah, well, a lot of those books are just structural formatting or, regulatory notes telling the cell when to read other books.
The filler stuff.
Basically. The exome, on the other hand, consists exclusively of the actual cooking instructions, the protein coding parts of your DNA.
Wait, so how much of the library is actually recipes?
It makes up only about 1.5% of your total genome.
Really? Just 1.5%.
Yeah, it's tiny. But, crucially, it is where the vast majority of known disease-causing mutations happen.
Okay, so we were scanning 1.1 million recipe books, but we are aggressively ignoring the formatting and only looking at the actual recipes. What exact typos are we looking for in there?
Well, the researchers were hunting for 2 main categories of incredibly rare variations. The first is called a missense variant.
Missense, okay.
Yeah, to stick with your analogy, a missense variant is like swapping salt for sugar in the recipe. The gene still produces a protein, like the dish still gets baked, but it might act completely differently or it might be structurally unstable. They found over 2.7 million of these missense variants.
Over 2 million swapped ingredients. Wow, and what's the second type?
The second is a PLOF, which stands for predicted loss of function.
Right.
So if a missense is a swapped ingredient, a PLF is like tearing a whole page out of the recipe book.
Oh, that sounds bad.
Yeah, the instruction stops abruptly. The resulting protein is either severely truncated, entirely broken, or just completely missing. And they found over 214,000 canonical PLFs across the cohort.
Okay, wait, so if a torn page is clearly disastrous for the recipe, What about the swapped ingredient? Could swapping salt for sugar, actually make the cake better by accident?
Usually, no. I mean, usually a missense variant just makes the resulting protein a little sluggish.
Okay.
If a gene is responsible for clearing out cholesterol, tearing the page out, so a PLOFS is bad. A missense variant in that same gene is typically also bad, just less severe. We call that a hypomorphic variant. But, as we are going to see, biology loves an exception. And sometimes a random typo does accidentally create a biological upgrade.
Okay, let me pause and make sure I am wrapping my head around the scale of this. For you listening, think about this. We are looking at the functional recipe book of DNA across over 1 million diverse people, hunting for torn pages and swapped ingredients that dictate our cholesterol levels.
Exactly.
Which makes me wonder, if you have a family history of heart disease, could there just be one single torn page pulling the strings? But wait. If we can spot these torn pages and swapped ingredients so clearly with modern tech, why are people still slipping through the cracks? Is the cellular machinery somehow misreading the pages that are perfectly intact?
That is exactly what happens. And it brings us to one of the most fascinating discoveries in the paper, because DNA doesn't just get read straight through by the cell.
Right, it's not a final draft.
Exactly. It has to be heavily edited first. When DNA is transcribed into RNA, it contains a bunch of filler text called introns. Those introns need to be edited or spliced out, leaving only the important instructions, the exons. The cell uses specific chemical flags in the genetic code to know exactly where to make the cut.
Like a molecular proofreader.
Yes. But the researchers used a highly advanced deep learning algorithm called Splice AI to hunt for what they call cryptic splice variants.
Cryptic splices? That sounds sneaky.
Very. These are incredibly sneaky DNA typos that do not look broken at all to a normal screening tool. The gene looks totally fine.
But it's not.
No. What the typo actually does is trick the cell's editing machinery into slicing the genetic instructions at the entirely wrong spot.
So the recipe clearly says, add a cup of flour, but the cryptic splice tricks the chef into editing it, so they literally read add a cup of flour.
Yes, exactly, a cup of flour. And you can imagine what that does to the cake.
A very crunchy cake.
Right. Splice AI acts as an advanced proofreader, catching these specific typographical disasters. A perfect example in the data is a variant in the APOA5 gene. It's what geneticists call a splice donor fifth base variant. For years, traditional clinical genetics just looked at this mutation and thought it was a harmless typo in the margins. It was not classified as a loss of function.
But Splice AI flagged it. So what is the actual mechanism there? What happens to the person walking around with that typo?
Well, Splice AI predicted it would ruin the editing process. And when they looked at the actual blood work of the real people who have this mutation, it acts exactly like a torn page.
Wow.
The APOA5 gene normally produces a protein that is crucial for activating lipoprotein lipase, which is what clears triglycerides from your blood.
Okay, so it clears the fat.
Right. But because the edit is wrong, the protein is mangled, it cannot activate the clearance mechanism, and the person experiences a massive, dangerous spike in their triglyceride levels.
Jeez.
And they found 15 of these cryptic splice variants across the genome that act as hidden master switches.
So to recap, we have been staring at perfectly intact genes, completely missing the fact that our own cells are slicing them up wrong. Meaning, if you are taking a standard genetic test right now, it is completely blind to these cryptic splices.
Pretty much.
But what about those swapped ingredients we talked about, the missense variants? You said earlier they usually just make the protein sluggish.
Usually, yes. That is the hypomorphic effect. But here is the big surprise from the data. About 13% of the time, the missense does the exact opposite of what you would expect a broken gene to do.
The opposite.
Yeah. We call this a hypermorphic variant.
Wait, hold on. If I have a gene that clears bad cholesterol and a mutation breaks it, my cholesterol goes up. Are you telling me a typo in that same gene can somehow make it clear cholesterol better?
That is exactly what I'm saying.
That defies logic. How does a random mutation accidentally create a biological superpower?
It sounds totally counterintuitive, but let's look at the actual mechanism using the LDLR gene.
Okay.
This gene produces the primary receptors on the surface of your liver cells that literally grab bad cholesterol out of your blood and clear it away.
Like little grabber arms.
Right. Now, if you have a PLFF that tears the page and breaks the LDLR gene, you don't have enough receptors. Your LDL goes sky high.
Right. Less clearance, more buildup. Simple.
Exactly. So standard logic says a missense typo would also damage the receptor and raise LDL. But they found a specific missense variant, LDLR PS L.
Okay.
It changes a single amino acid, a serine, to a leucine. Instead of breaking the receptor, this specific structural change actually stabilizes it. It supercharges the receptor's ability to bind to cholesterol or recycles it back to the cell surface way faster.
So it is literally working overtime.
Yes, people with this incredibly rare swapped ingredient have strongly decreased LDL levels. Their bodies are hyper-efficient at clearing bad cholesterol.
Oh, wow.
Conversely, they found a variant in the APOB gene that acts as a gain of function and unexpectedly raises LDL. And here's where our traditional algorithms completely faceplant.
Because they just assume it's bad.
Exactly. They're programmed to see a typo and automatically flag it as broken or deleterious. They're totally blind to the fact that a typo might actually be an upgrade.
That is wild. It's like finding out the spell checker on your computer was programmed by someone who only understands half the language. It flags everything as an error, even if it is just a more advanced vocabulary word.
That's a great way to put it.
Which makes you wonder, right? How many supercharged risky genes are slipping under the radar right now because the algorithms just assume they are broken?
Quite a few, unfortunately.
But what happens when we start looking at how these genes are inherited? I mean, we get one copy of a gene from each parent. If our algorithms are missing these upgrades, how are they handling the math when you inherit two typos? Are we just basically adding them together?
So that is a standard approach, yeah. Standard genetic risk models assume an additive effect.
Okay.
If inheriting one bad copy of a gene raises your cholesterol by 10 points, the additive model assumes inheriting two bad copies, so one from each parent, will just raise it by 20 points.
Which seems entirely logical. 1 + 1 = 2.
But biology rarely sticks to simple arithmetic. The researchers in this study explicitly modeled for recessive effects. They wanted to know what happens specifically when a person inherits two identical copies of a rare variant.
And what did they find?
They found 109 genetic associations where having two copies triggers a radically different, massively compounding biological effect.
Wait, in what universe does having two defective genes suddenly cause a radically different outcome than having one? Shouldn't it just be double the problem?
Let me give you the clearest example from the paper, which involves the HBB gene, specifically a variant called PE7V. You probably know this mutation by its clinical result. Inheriting two copies of this exact variant causes sickle cell anemia.
Oh, okay. But what does a disease of the red blood cells have to do with my cholesterol levels?
That is the amazing discovery here. If you look at this variant through a standard additive model, meaning you just look at people with one copy who have sickle cell trait, it has incredibly weak, almost unnoticeable effects on cholesterol.
So barely a blip.
Barely a blip. But if you look at it recessively, so people with two copies and full sickle cell anemia, it causes massive, drastic drops in both total cholesterol and LDL.
Wait, what is the mechanism there? Why does the cholesterol just vanish?
Because the body is under profound systemic stress. In sickle cell anemia, red blood cells are constantly deforming and being destroyed. The body has to work in absolute overdrive to continually manufacture new red blood cells to replace them.
Oh, I see.
And that manufacturing process requires a massive amount of cholesterol to build new cell membranes. The drastic drop in blood lipids isn't a superpower. It's a byproduct of the body rapidly consuming its own cholesterol pools just to survive the high turnover of red blood cells.
Wow. It is literally draining the tank just to keep the engine running.
Exactly.
And, because sickle cell is predominantly found in people of African descent, this brings us right back to why that 20% non-European diversity in the study cohort is so critical.
It is the absolute cornerstone of why this study matters. By including over 238,000 diverse genomes, the researchers discovered 130 lipid-associated alleles that were significant only in non-European populations.
Only in non-European?
Yes. These are massive genetic master switches that have been entirely missed by a decade of Eurocentric science.
Okay, let's dig into that. Give me an example of a master switch we have just been ignoring.
Let's look at the PCSK9 gene. There is a specific stop-gain variant, so a torn page, called PC679X. In European populations, this variant is practically a ghost. It occurs in less than 0.1% of people.
So practically nobody.
Right. But in African-like populations, it is highly prevalent, appearing in nearly 1% of individuals.
Okay, and mechanically, what does breaking the PCSK9 gene actually do?
Well, the PCSK9 protein normally acts like a garbage truck for your liver's cholesterol receptors. It binds to those LDLR receptors we talked about earlier and drags them away to be destroyed.
So it's clearing out the clears.
Exactly. So if you have a torn page in the PCSK9 gene, you do not produce those garbage trucks.
which means the liver gets to keep its cholesterol receptors.
Yes, the receptors stay on the surface, pulling bad cholesterol out of the blood continuously. If you have this variant, it consistently and massively lowers your LDL cholesterol. It is highly protective.
But because it is virtually absent in Europeans, previous clinical studies just didn't have the statistical power to definitively prove its effect.
Precisely.
Wait, so let me get this straight. If a highly protective genetic variant is found in 1% of people with African ancestry, but is basically zero in Europeans, and historically all our clinical risk scores and diagnostic tools are built exclusively on European data, I mean, we've been completely blind to this. Are we fundamentally misdiagnosing diverse populations because our baseline is just wrong?
We absolutely haven't. And the crucial takeaway here is understanding the difference between allele frequency and biological effect size.
Okay, break that down for me.
The allele frequency, which is how common a mutation is, differs wildly between ancestries. But the biological effect size, meaning what the mutation actually physically does to your liver or your blood, is shared entirely across ancestries.
So if I had it, it would do the same thing.
Exactly. If a European person somehow inherited that PCSK9 variant, it would destroy their garbage trucks and lower their LDL just as much. The biology is universal, but the distribution is not. Discovering these ancestry-enriched variants is a colossal leap for health equity.
So if I am taking a genetic test that is only based on European data, it might not even reflect my actual genetic reality.
Unfortunately, yes.
Which leads me to the ultimate question. How do we fix the tests? Because we're not just dealing with abstract data points here. We're talking about real people with conditions like familial hypercholesterolemia or FH.
Right, which is very serious.
For you listening, FH is a genetic condition that causes dangerously high LDL levels right from birth, leading to severe heart attacks in people in their 30s or 40s.
And to diagnose and treat FH, doctors rely on clinical databases. The gold standard is a database called ClinVar. Think of ClinVar as the ultimate, continuously updated medical encyclopedia of genetic diseases.
Okay.
Geneticists submit variants to it, classifying them as either benign, pathogenic, or a very frustrating category called variants of uncertain significance, or VUS.
Right. A VUS is basically the doctor throwing their hands up and saying, you know, you have a typo in your DNA, but we have absolutely no idea if it's going to cause a heart attack or do nothing at all.
Exactly. And what the researchers did here was revolutionary. They took 9,620 FH variants currently listed in the ClinVar encyclopedia and cross-checked them against the actual real-world lipid data of their 1 million participants.
They tested the encyclopedia.
They essentially asked, does the medical encyclopedia match human reality?
And did it.
Not even close. They found a shocking number of necessary reclassifications. Variants currently listed in ClinVar as benign or sitting in that frustrating limbo of uncertain significance actually have massive, highly pathogenic effects on LDL in real life.
Wow. Do we have specific examples of what got upgraded to a threat?
Absolutely. There is a variant called LDLR PH327Y, which is enriched heavily in South Asian populations. There's another called LDLR PL456P, enriched in African populations. In ClinVar, these were flagged as VUS or inconclusive.
So a doctor wouldn't worry.
Right. A cardiologist seeing these on a patient's chart wouldn't necessarily know to prescribe aggressive, life-saving lipid-lowering drugs like statins or PCSK9 inhibitors.
Because the database said, we're not sure. So people with these variants were essentially walking around with undiagnosed ticking time bombs.
Precisely. But this new population-level data proves undeniably that these specific variants act exactly like established highly pathogenic variants causing FH. They cause a massive, dangerous spike in LDL. The encyclopedia was simply lacking the diverse data to make the call.
That is just terrifying and amazing at the same time.
It is. And the researchers didn't stop at just measuring blood cholesterol. They took all 800 significant lipid variants they found and cross-referenced them with hard, undeniable clinical outcomes, coronary artery disease or CAD. They wanted to know if the people with these mutations actually suffered heart attacks.
So bridging the gap from just a blood test marker to actual cardiovascular disease.
Right. And through this analysis, they identified 5 specific genes directly tied to CAD risk. But the absolute biggest breakthrough, the one pharmaceutical companies are looking at very closely right now, was a gene called RORC.
RORC. Okay, what does RORC actually do? If we are talking about heart disease, is RORC normally causing inflammation in my arteries? Why is my body producing it in the first place?
Well, RORC is a critical transcription factor in your immune system. It plays a major role in the differentiation of a specific type of immune cell called Th17 cells. which are heavily involved in driving inflammation.
Oh, immune cells. Okay.
Now, in your blood vessels, chronic inflammation is the spark that accelerates plaque buildup. It drives the accumulation of lipids into the artery walls.
Ah, so it is immunometabolism. The immune system is directly worsening the cholesterol problem.
Exactly. So what the researchers found is that having a broken RORC gene, so a naturally occurring P-alpha-F variant that tears the page out, lowers your systemic inflammation and significantly protects you from coronary artery disease.
Really?
Yes. It is directly associated with lower LDL and a decreased risk of heart attacks. Prior studies in mice hinted that suppressing this gene improved metabolic health. But this massive human data set proves definitively that a naturally occurring loss of function in RORC is cardiovascularly protective in human beings.
So pharmaceutical companies must be drooling over this. If they can design a highly targeted drug to silence the RORC gene, basically creating a synthetic torn page, a synthetic PLF, they can artificially reduce that inflammation, lower LDL, and prevent heart attacks in the general population.
Yeah, it is literally a treasure map for the next blockbuster cardiovascular drug. It is the absolute pinnacle of genomic medicine. You locate a rare, naturally occurring mutation that protects an individual against a disease, and you reverse engineer a drug to mimic that exact protection for everyone else.
That is incredible. I want to pause here and sincerely thank you for guiding us through all of this. This has been an incredibly dense roadmap of our DNA.
It's been a pleasure.
We have seen how looking at 1.1 million diverse exomes didn't just find a few new typos. It proved how algorithms like Splice AI can uncover cryptic splices that trick our cells, how hypermorphic variants defy logic and supercharge our biology, how deeply crucial our ancestral background is to our true risk profiles, and how this influx of data is literally rewriting the medical encyclopedias on heart disease right now.
It really is a profound paradigm shift in how we approach targeted personalized medicine.
And for you listening, I know the sheer volume of genetic data and mechanistic pathways we just covered can feel totally overwhelming. It can feel like alphabet soup. But remember, this knowledge, these microscopic, obscure typos, the missense variants and the cryptic splices, this is the shortcut to true personalized medicine. This isn't just abstract biological data. It is the actual instruction manual to you.
That's beautifully said.
Thank you. I want to leave you with a final thought to ponder as we wrap up. We just saw how clinical databases like ClinVar are being continually forced to update and correct themselves based on these massive multi-ancestral studies. That means the very definition of what is considered healthy versus what is categorized as pathogenic is literally shifting under our feet as we speak. The genetic test you take 10 years from now might give you entirely different, potentially life-saving answers than the exact same biological test taken today. Are we prepared for an era of medicine where our physiological reality updates as frequently and as radically as the software on our phones? Keep digging, keep questioning, and we'll see you on the next Deep Dive.